All right, hello again, everyone. This week we're going to be talking about not only cell respiration, but also photosynthesis using Elodea plants. And so if you recall from our previous experiments with the fish, we talked about cell respiration, and we talked about how water, when it reacts with carbon dioxide, becomes acidic, right? Well, now we're talking about a plant. So obviously a plant can do something we cannot. It undergoes photosynthesis. And as we've been learning, photosynthesis actually takes carbon dioxide out of the environment and uses that carbon dioxide as a carbon source to make sugars, right? That's how a plant grows and how a plant stores its energy. But this gets a little bit more complex because what you have to remember is that plants do cellular respiration too. Let me say that again. Plants do cellular respiration too. Think about it. What do you need for photosynthesis? Well, carbon dioxide, obviously. We need water. Hopefully you know why you need those two things now. But you also need light. Is a plant alive at night? Absolutely. But it can't do photosynthesis. What it's doing is cellular respiration and using those sugars that it has stored up. So a plant does both cellular respiration and photosynthesis. And it does cellular respiration all the time, light and dark, and photosynthesis only in the light. So we need to account for two of those things. So let's think about this for a second. So here's an aquatic plant. This is our Elodea in the water. So this plant we know is undergoing cellular respiration for sure. It's alive. So what happens when something undergoes cellular respiration? So let's focus here and think about this for a second. This is in water that is at a pH of 7.2. It has phenolphthalein in it. But we don't see pink because remember, our phenolphthalein only turns pink, it has that color change at a pH of 8.2. So if this plant, as we know, is undergoing cellular respiration, that pH is going down because the cellular respiration is producing carbon dioxide just like with the fish, therefore causing the pH of this water here, causing the pH to go down due to cellular respiration. Now, if this is in light, then it's also doing photosynthesis. And photosynthesis is pulling carbon dioxide out of the water. And so photosynthesis would actually be removing carbon dioxide, making the pH go up. And so if we have cellular respiration, and then we have photosynthesis, can I draw backwards? Which direction does that P go? I don't know, photosynthesis. Good enough. Then photosynthesis going up. If we are doing more photosynthesis than cellular respiration, that pH will come up towards 8.2. If we are doing less photosynthesis than cellular respiration, that pH will get further from 7.2. So this is where your designed experiment comes in. You're going to design an experiment that we're going to do where you're testing, you're going to create a hypothesis about what you think will cause more or less photosynthesis in the Elodea plant. And so we want to then compare. We're going to have several beakers. We're going to have a control beaker that has no plant. We're going to have a beaker that's in the dark, only doing cellular respiration, and then beakers that are in various conditions that you've decided. And then we are going to use titration, just like with the cellular respiration lab, to see how much it takes to get to that color change. Only this time we're coming up to the color change of pink. And so if something's doing a lot of photosynthesis, it's not going to need as much titration to become pink. If something is doing very little photosynthesis, it's going to be a long ways away from 8.2 and it will need more titration. That's our analysis. Right? But again, we have some controlled variables. We want to make sure we're using the same amount of plant. With the fish, we weighed the fish. For the plant, we're going to use volume. So I have a beaker here. And with this beaker, I know, remember, use your meniscus, get at eye level. This beaker is at 40 mils right now. So then I am going to insert my plant. And I can see that that water level has risen to 41.4 mils. So this is 1.4 milliliters of plant. So I would want to make sure that I'm getting the same amount of plant for all my different groups. I would then have all my beakers with the same amount of plant in them. We would let that go in under whatever conditions you've decided to test for a half an hour. And then at the end, we are going to take those beakers and we are going to titrate to see how much volume it takes to bring us to 8.2 where it turns pink. So let's think about this for a second. We are coming up in pH now. So do I want to use my acid like I did for the fish? Well, acid is going to bring the pH down. This time I need to go up to 8.2. So I'm not going to use the acid. I am going to use the base to bring the pH up. And then we're going to use our standard titration method. Taking a look at this ahead of time. This is at 32.5 mils currently. And then I am going to titrate. All right, so as we're titrating, get my control beaker in here again. This one you really want to slow down because when it goes from clear to pink, there is no gradual change. It's just going to start to turn pink, and that pink is very subtle. Whereas going the other direction, you could see it was getting less pink, and then you could find that spot right when it turns clear. Here, we want to be really careful, and you can start to see, I don't know if you can zoom in there a little bit, but you can see that there's a different color kind of in that right there. It's starting to be a little bit pink. And so as long as you're consistent in titrating all of them, we can go a little bit further and kind of see if this gets a little bit more pink, but you want to make sure you are really careful and get right to that subtle pink color because that's how much it took to get to 8.2. But I'm just going to leave it at that at the moment. So now I would just find the final volume. So we're now at 48.5. 48.5 minus 30.5, we know that that took 18 mils of titration. And I would just compare this with all my different groups, and that is going to tell me which ones did the most photosynthesis, because the ones that took the least titration must have done more photosynthesis. They were already closer to 8.2 than the ones that did the least photosynthesis. All right, thank you.